antioxidant activity. Kranti sir, sharing is not there.
Sharing, sir. Give me 1 minute, sir.
Oh.
The link is going live, sir. So because of that, it was blocked. Yes, sir. Give me 1 minute, sir. 1 minute. The link is going live, sir. Yes, sir. Give me 1 minute, sir. 1 minute. The link is going live, sir. Yes, sir.
So, today's seminar concept for how to identify the individual components present in the given mixture and likely to produce antioxidant activity instead of a total extract showing the maximum activity when compared to the standard drugs. Because of natural products wise, there is no standard should be compared with one chemical agent. That's why we want to identify the chemical agent which is having the maximum antioxidant activity when compared with the standard. Then what is the threat of oxidative stress? The main problem is the cellular homeostasis will be disturbed due to the reactive oxygen species develop. When ROS levels overwhelm the endogenous cellular defenses, oxidative stress ensues, accelerating the aging and driving the pathology of cancer, diabetes, and cardiovascular disease. Thereby, the KEAP1 repression is inhibited and activating the NRF2 transcription factor, thereby induce antioxidant genes and producing the antioxidant activity. Next slide.
Sir, please, sir, if you are watching through the system, please mute it, sir. Echo వస్తుంది సార్ మీ సిస్టంలో ఉంటే కనుక సిస్టమ్
Okay. I muted.
Yes.
Okay, please share that. The next one is then okay, sir?
Yes, sir. It is okay now, sir. It is okay.
Okay, sir. Then the types of free radicals generated generally, the two types. One is oxygen-centered species. Second one is nitrogen-containing the species. When the nitrogen-containing species, the examples are nitrogen dioxide, oxygen-containing species, all are known one, superoxide anion, hydroxyl radical, singlet oxygen, hydrogen peroxide, and peroxyl radicals. These radicals are unstable molecules and having the unpaired electron means that odd electron. Thereby broadly categorized into the two categories. Next slide, please.
Oxygen-containing species, all are known superoxide anion, hydroxyl radical, singlet oxygen, hydrogen peroxide, and the peroxyl radical. It prioritize for unstable molecules on size where unpaired electrons meet that odd electrons in complete nitrogen structure. When the neighborhood by broadly transposed to one two surfaces, just closely, and the species are only two surfaces.
Then the chemical defenders, which are acting in that antioxidant activity, generally lipid peroxidation chain reactions will be happening. The lipid peroxide chain reactions will be producing a respective uh, reactive species. They are called as the reactive oxygen species. Thereby, the antioxidants protect the agents. Antioxidants protect the agents that stabilize critical cellular components like lipids, proteins, DNA by neutralizing reactive oxygen species. Thereby, they donate an electron or hydrogen atom to the radical, safely converting absorbing the oxidative burden without becoming dangerously reactive themselves. Thereby, the oxidants donates either electron or hydrogen to the reactive species and neutralizing the chain reaction, lipid peroxidation chain reactions especially, and maintaining the homeostasis. Next slide. Then, how many types of antioxidants are there in the literature on the basis of functional taxonomy? These are two varieties, primary antioxidants, secondary antioxidants. Then mode of action, on the basis of mode of action, they are segregated into two categories. By direct interception, thereby reacts with the lipid radicals to convert them into stable products. They are called as the primary antioxidants. The biological examples are SOD, catalase, glutathione, peroxidase. The botanical examples are flavonoids, catechins, carotenoids, etc. Similarly, on the basis of mode of action, they are classified into two a second variety called as the secondary antioxidants. The regeneration and capture is the mechanism of action. They stop the chain reaction, which we have discussed in the previous slide, and maintains the antioxidant system. The examples of biological are glutathione reductase, G6PD. The synthetic examples are BHA and BHT. Next slide, please. Next slide, please. Next slide. The antioxidants further classified on the basis of the scavenging activity into two types, enzymatic antioxidants, non-enzymatic antioxidants. Thereby, the enzymatic antioxidants furtherly classified into two types, primary enzymes, examples are SOD and catalase, secondary enzymes are glutathione reductase. Similarly, non-enzymatic antioxidants classified into three varieties, vitamins and their derivatives. Examples are vitamin A, C, and E. Second example, cofactors. These are nothing but coenzymes. And chemicals are phenolic acids, ferulic acid, gallic acid, etc. Next. The in vitro evolution happened by two types of the mechanisms to carry out the total antioxidant capacity, which measures the aggregate free radicals scavenged by a test solution. One is called as the hydrogen atom transfer, and second one is called as the single electron transfer. In case of hydrogen atom transfer, the molecule of the chemical agent donates the hydrogen atom to the respective substrate where the free radicals are generated and neutralizing the reactive species. In case of single electron transfer, the chemical compound issues the one electron, thereby neutralizes the negative charged species into positive charged species. Thereby, these two mechanisms are effectively produces the antioxidant activity where you can measure the total antioxidant capacity for the given molecule. Then coming to the next one, the single electron transfer spectrometric shifts. Next slide, sir. There is four methods are available generally. One is, next slide, please. There are single electron transfer consisting the four basic methods. One is DPPH, which makes the stable radical reduced to non-radical. ABTS, chromophore reduction. FRAP, ferric iron complex reduced to ferrous form. And cuprac, that is the cupric neocuprione complex reduction. This DPPH method, the color changed from some radical formation, thereby ABTS, the blue color or green color converting to colorless, and the FRAP, yellow color converting into blue color, and ORAC system decrease or increasing the fluorescence, and cuprac method light blue or green color to yellow orange. By using the either spectrophotometric method or UV visible spectrophotometry or fluorometric methods, we can able to measure the total antioxidant capacity of the given molecule or extract. Then next one, the HAT versus C in the DPPHSA. Sir, please put the previous slide, sir. Ranti, sir.
Sir, you want me to change it, sir?
Please go to the previous slide.
Radical formation thereby ABCA, the blue color or green color converting to colorless, and the FRAP, yellow color converting to blue color, and ORAC system decrease or
Okay.
fluorescence.
No, previous one, sir. HAT versus set mechanisms. The HAT mechanism, the reaction mechanism is quenches free radicals by donating hydrogen. Set mechanism is transfer an electron to reduce the oxidant tracked by color change. Please, previous method, sir. Previous slide, sir. Yes, sir. Then HAT mechanism target relevance is peroxyl radical, physiologically highly relevant, whereas set mechanism is fluorescent or colored probes, analytical proxies. Environmental dependence is pH dependent by the HAT mechanism, whereas set mechanism is highly pH dependent. Reaction kinetics rapid, completed in seconds to minutes, whereas set mechanism is very slower, relies on percent decrease in product rather than kinetic decay. Primary assays used for the by HAT mechanism is Varrock, Lippic, and trap mechanism, whereas the set mechanism is that means single electron transfer mechanism follows the FRAP technique, TEAC method, or cupric reduction method. So that is the CUPRAC method.
Sir,
Next slide.
sir, yes, sir.
Sir.
Sir, give me 1 second, sir. In YouTube it is coming as echo. ఒకసారి దాన్ని క్లోజ్ చేసి మీకు మళ్ళీ మళ్ళీ షేర్ చేస్తాను సార్. 1 second.
Okay.
Then, past mechanism, target segment is corruption radical, physiological holiday element, whereas second mechanism is fluorescent or color probe, and the
Sir, now it is okay, sir.
Okay, sir.
Yes, sir. You need me to change this slide, sir?
Next slide, please.
Yes.
Then the universal SA method used for total antioxidant capacity is the DPPH SA method, where we are using the 2,2-diphenyl 1-picryl hydrazole remains the standard for unified total capacity assessment. Thereby, the color changes will give the system. You can observe here, in case of the absorbance spectrum, the absorbance is drop. The absorbance is drop. That indicates that the color changed from deep color to a less intense color, which indicates the activity. Then how color is shaded, how the deep color will be shaded, degree of deepening is reduced, that is having the maximum antioxidant activity. Next slide, sir. There is a simple mapping I made here, that is the ORC system, oxygen radical absorbance capacity, which measures fluorescence loss when an azo-initiator destroys a fluorescent molecule. The instrument used is the fluorimetry, in which the color is decreased, that means not color, fluorescence is decreased. That's why you want to use the fluorimetry. In case of the FRAP, ferric reducing antioxidant power, that will be carried out at low pH values, in which the yellow color will be deepened to blue color. Because of the color development, we want to use the UV visible better diode array detector. The third method is ABTS or TEAC method, which monitors the fading of the blue-green chromophore upon oxidation introduction. The instrument required is the diode array spectrophotometer, where the color change from dark blue to the dark blue or dark green to the colorless molecules. Then cupric ion reducing capacity evaluates reduction of cupric neocuprion complex to cuprous, that is the neocuprion. In the presence of chelating agent, instrument calorimeter is sufficient for this one. In case of the cupric method, light blue or green color change it to the yellow-orange color or yellow color. This is a simple comparison of the spectrophotometric methods used for the total antioxidant capacity. Next slide, please. Here you can see the how to carry out the total antioxidant capacity essay. Similarly, what is the use of AI in case of the antioxidant activity? So there is the four steps are there. Step one, total capacity, measuring the synergetic redox interactions. Step two, in vitro assays, that is the mechanism based one. Standardizing the hat and set radical scavenging methods. Then chromatographic profiling, molecular view, separating complex phytochemical matrices. On step 4, using the AI, you can able to predict by using the software called as the SMILES strings. What is the SMILES strings? It is the simplified molecular input line entry system, which is the a compact structures are produced by the SMILE system. We can able to easily understand which structure of the molecules are available in the given mixture of the component and how that is giving the antioxidant activity. Next slide, please. So in case of the spectrophotometric and fluorometric screening methods, the core methodology involved is introducing radical probe to botanical sample, incubate under strictly controlled temperature parameters, measure absolute optical absorbance or fluorescence decay. This is it required the some cell-free environment system utilized to rapidly measure the fundamental ability of compounds to scavenge the free radicals. Various accessories present in the instrumentation I would like to show here. See here the design, the 96 well plate, that is the reading chamber, and UV visible spectrophotometer generally used. There the detector is the emission detector, that is the photomultiplier tube, then monochromator and reading chamber followed by data output interface to the computer system, and there is the automated plate transporter is also there. This is the instrumentation inside accessories available when we carried out the spectrophotometric based TASC system. Next slide, please. What is the previous in vitro assays used for the antioxidant method, then compared with the present chromatograph technique? The total antioxidant capacity is a simple as active, but it treats complex when using the natural products. Because of the natural products means it is the black box technology. What is what is present in the extract we don't know. That's why that is called as the black box technique. In case of the in vitro assays, it provides the total capacity, functional activity, radical scavenging kinetics, but unable to provide the identification of the specific molecules, revealing structural synergism. In case of advanced chromatography, we can able to provide the molecular identity, separation of complex mixtures, precise quantification, that is by using the mass spectrum, especially mass spectroscopy. But the proving actual functional biological activity if they are not paid. You can see here in the chromatogram also we are showing I'm keeping the chromatogram on the instrumentation. We can able to identify the components as the HPLC peaks by using the present chromatographic system. Next slide. When using the HPLC MS by MS, we will get a better result for the chromatographic system. Thereby, you can able to separate the complex mixers using the HPLC MS by HPLC system to this above the complex mixers into individual parts. By using the mass spectrum, it provides the definitive molecular identification via precise mass to charge ratios and precursor to product ion transition. You can see in that figure, which is surrounded by the circle. The peak is identified as the chlorogenic acid. That is the advantage of using the present chromatographic technique instead of only the old models of only in vitro assays of the system. So by using these spectroscopic methods, you can able to identify the compounds. You can able to separate and identify the compounds. You can able to understand which compound is present, which compound is giving the antioxidant activity can be predicted by using this spectroscopic technology. Next slide. Then see here the mass spectroscopy. This is all or known one. Okay. The liquid separation sample exists from the column with high pressure into LC column, where generally electrospray ionization nozzle converts liquid sample into a charged mass. We are using the electrospray ionization. Later, there is the mass analyzers are there. Depending upon the instrumentation, different mass analyzers are used, either quadrupole, triple quadrupole ion trapping system, or cyclotron system, or the multifunctional hyphenated mass spectroscopic techniques like time of flight and quadrupole and different methods or different varieties of the mass analyzers, where the magnetic fields selectively filter and accelerate the molecular ions. Finally, we are getting the detection mass spectrum, which gives the graph precise mass to charge ratio to generate an unmistakable chemical fingerprint. This is the advantage of using the HPLC with the mass spectrum to detect and identify and quantify a given compound, a given natural product which is having the different kinds of the chemical components. Next slide, please. There is another spectroscopy is developed by the scientist called as the kinetic EPR spectroscopy. EPR stands for electron paramagnetic resonance spectroscopy. It is presently using as a traditional assays instead of traditional assays. There we are using the colors. Here we are using the spectroscopic technique. Its spectroscopy offers direct kinetic measurements of unpaired electrons by using online pulse reagent injection. Thereby, researchers easy to know the exact mechanistic rate of radical scavenging as it happening in the biological system. That is the most advantage of the kinetic EPR spectroscopy. Next slide, please. So you can see here what is the advantage of using the HPLC system, especially UPLC MSR also. The peaks are very nicely separated, but you want to select the column, very nice column. When the scientists are using 5 micron particle packing, broad overlapping peak resolution is obtained. The runtime is also takes the more than the 30 minutes. Whereas using the micron particle of packing, that is the sub 2 micron particle packing, a smaller particle size generally increases the peak resolution, sensitivity, as well as the speed of the analysis. Thereby runtime reduced to less than 10 minutes. There you can see that in the standard HPLC with the large particles, overlapping peaks are there. When using the less particle size of the columns, using the especially EPLC, you are getting a good resolution separation of the peaks. That is the advantage of the present spectroscopic technique in case of a total antioxidant capacity measurement. Next slide. Then not only HPLC, in case of total antioxidant capacity, we can able to use the GC also, because of all the samples may not be volatile, all the samples may be volatile. So samples which are volatile, samples which are highly volatile, use the GC system along with that means hyphenated with the mass spectroscopy, also obtained a but a nice identification of the chemical compounds which are having the total antioxidant capacity. Liquid extracts are vaporized and transitioned to gas, allowing the system to separate and identify highly volatile antioxidant compounds, glucans, and essential oils that liquid chromatography which cannot capture. That means HPLC cannot capture. Similarly, the natural products having the very advantage by using the GC to evaluate components present in the natural products which are having the antioxidant capacity by using the GC as well as GCMS also. Next slide, please. Next slide. Okay. Previous slide. Analytical technology used are HPLC UV dot, HPLC EPLCMS, GCMS, and kinetic EPR. These are the four variety of the spectroscopic techniques used for total antioxidant capacity framework. Then primary target matrices are that is the HPLC UV dot is for generally routinely used for the flavonoids, whereas HPLC UPLS MS complex non-volatile phytochemicals, GCMS for the volatile compounds and essential oils, whereas the kinetic EPR is the direct unpaired electrons, compounds containing the direct unpaired electrons. Then coming to the speed of the work, HPLC dot is the moderate, HPLC UPLC MS is the high, GCMS is also moderate, kinetic EPR used for the real-time analysis. You can able to use it for the real-time analysis. Then resolution capability, HPLC dot is the moderate, UPLC-MS is the absolute and very precise ambiguous ratios. GCMS is phase-dependent gas profiling, and kinetic APR is a kinetic mechanism confirmation studies which has been used. Next slide. Then coming to the examples of the natural products which are having the antioxidants, applying the studied by HPLCMS, UPLCMS, GCMS like that. The best examples are chrysanthemum indici floss. This is generally in Telugu it is called as the chamanti. There 36 compounds are identified by HPLC, quadrupole time of flight mass analyzer. The major actors are isochlorogenic acids and iriodactyls. Whereas in the guava leaves, CDM guava species, low oxidation states yield vast flavonols and ellagic acid derivatives extracted via ultra-assisted extraction method. Similarly, all known leaf is the Mentha piperita, that is the peppermint leaf, from which the HPLC analysis confirms significant higher polyphenolic yield and FRAP and DPPH activity compared to the stevia. Stevia in the sense it is the sweet tulsi, that is called as the sweet tulsi or madhu tulsi. There, by using the HPLCMS or UPLCMS, the plants are already studied for the antioxidant capacity. Next slide. Not only the plant products of the leaves and other things, then agro-industrial byproducts. Agro-industrial byproduct. That means waste products of the industry, waste materials of the industry will likely to give the similar process. See the almond skins. Generally, we are removing the almond skins. By using the microwave assisted extraction isolates extreme concentration of flavonols and camphorol in the guava cultivar. Similarly, potato peels, Solanum tuberosum, The bluebell peel exhibits peak FRAP-ORAC activity driven by caffeoyl caffeoylphenic acid isomers. Then chestnut shells, that is called as the Castrea sativa, subcritical water extraction yields rich pyrogalal and protocatechuic acid. This is the generally we are wasting this waste from the even though from home also. Generally, when we are using the potato, we are removing the peels. When we are eating the almond, soaking the almond and remove the top shell of the almond, skin of the almond. Generally, shells all or one shell will be removed. But even though in this waste also there is lot of flavonoids are there will be studied by using the HPLC or UPLC MS systems. Next one, the pigmented fruits and seeds also having the antioxidants like anthocyanins and phenolic acids which act as intense free radical scavengers. The best example is the black goji. The scientific name is Lycium rutheniacum. Petunidin-based derivatives are identified from the total anthocyanins. The instrument applied is the HPLC dot. It reveals the strong negative correlation between the acylated anthocyanins and FRAP activity. Similarly, all are known fruits, small berries, HPLC HRMS confirms the black currants and blueberries dominate total flavonoid counts compared to 11 other regional berries. Then apple seeds. Apple seeds, all are generally we are thrown out the apple seeds. The HPLC PDA mapping unexpectedly reveals that Honeycrisp seeds rich in fluoridizin and heparin drastically outperform the apple's peel or flesh in total phenolic content. So there is no waste in the either leaves, flowers, and seeds, as well as the fruits, as well as the shells of the any seeds are also. So that is the advantage of taking the natural products to get the maximum antioxidants from their sources. The next slide, please. Not only the flowers, leaves, and other things, we will also getting the tea also. Plant tea materials are also will provide the good functions capacity of the antioxidants, which are generally called as the medicinal beverages. The best example is DBT, Dujang brick tea. The HPL's FRAP technique identified 11 compounds, isolating chlorogenic acid as the primary radical scavenger. Similarly, Actinidea arguta, this is called as the hardy kiwi leaves. Solvents is the matter here. The ethanol generally used as the extraction to maximize hydroxycinemic acid yield over aqueous steeping, perfectly protecting IEC6 cell lines from reactive oxygen species. Similarly, all are known one green tea, black tea, fruit tea, in which the FRAP RF HPL's FRAP technique prove green tea consistently retains higher antioxidant density than black or fruit of the forest trees. Anyhow, which is having the more, which is having the less maybe, but all the teas are having green tea, black tea, fruit tea, having their own benefits in terms of the total phenolic content, but as a total oxidant capacity. So natural products will serve as the excellent source as the antioxidants to maintaining the cell homeostasis. Then not only the plant materials, teas, seeds and other things, the fermentation and hydrolysis techniques also provide the excellent antioxidants which has proved by the literature also. Then, by using this fermentation technique, scientists are one of the example I will like to quote here. By solid state fermentation, Angelica dihurica fermented with Erotium cristatum fungus. Then, after fermentation, they used the HPLC PDA triple TOF MS by MS reveals the creation of 12 unique new antioxidants. These are polyketides, indole alkaloids, which are not present in the unfermented route. So, fermentation process also develops the active, bioactive antioxidants from the natural sources. Similarly, hydrolysis. All are known on brown rice in present days. Take the germinated brown rice and carried out the acid hydrolysis as well as the basic hydrolysis. Scientists are identified the basic hydrolysis having the better antioxidants whereby carried out ABTS and FRAP activity, whereas acid hydrolysis yields minor increasing in the DPPH method antioxidants. Anyhow, either the acid hydrolysis, basic hydrolysis, in this case, base hydrolysis will provide the maximum antioxidants of the molecules in case of the rice are also, brown rice are also. So the fermentation and hydrolysis techniques also useful to generate the antioxidants from the raw sources. Next slide, please. Then another advantage of present spectroscopy, we can able to study the structure activity relationship. You can able to study the structure activity relationship. By the molecule may act as either HAT or ZSA method. Anyhow, here you can see in the left side, there is the glycosyl moieties are there which are not required for the activity, clearly stated by the spectroscopic methods and antioxidant assay methods. Then in case of the top right side top corner, methoxy substituents at ortho OH thereby boost the both HAT and SCT activity via pi conjugation system. Similarly, in the bottom of the molecule, I mentioned that 2-6-dihydroxy structure provides superior resonance stabilization to the molecule, thereby maximizing antioxidant potential over a 2-4-dihydroxy arrangement. So this type of the structure activity relationship also useful to study by using the total antioxidant capacity with the help of or with the aid of the chromatographic technique where the components are identified. Individual components were identified. Next slide, please. Then what is the integration in between the total work involved in the antioxidant capacity by combining with the new chromatographic techniques? First one is raw botanical matrix undergoes the optimized extraction, screening for capacity, use either the DPPH, FRAP, ORAC and any other screening methods. Does it work or not? We don't know. Then yields the only total antioxidant capacity only. But by using the fingerprint analysis, using the HPLC or UPLC, similarly using the mass, especially HPLC and UPLC, it gives the chromatographic resolution. It gives the chromatographic resolution.
Sir. Sir, yes, sir.
Okay?
Yes, sir.
Okay, after separation by chromatographic technique
Sir,
of the all the,
sir, we have already guest in the waiting room, sir.
just 2 minutes it will be complete. Okay. So thereby the components will be separated out and using the MS technique, either the mass analyzer, HRMS, we can able to quantify the molecules. Their capacity plus identity will give the validated biological efficacy. That is the integration of the total antioxidant capacity to the respective chromatographic technique in present days useful for carrying out the antioxidant activity. Finally, I would like to show the smiles how it will be useful to predict the active prediction from the inactive prediction molecules. So by using the smiles software, by using the data software, that is the database system, from the given mixture of the components, it will be they are differentiated how many active components are there, how many inactive components are there, from which we can able to predict the antioxidant capacity of the plant materials or either the material given for the analysis. Then next slide, next one. So finally, by using the different softwares, especially Tanimoto network system, the mapping structural similarity using the Tanimoto system, which identifies the different molecules are favorable from the a given pharmacophore. Suppose in the given chrysin, there is the different compounds are identified like tactochrysine, trihydroxyflavone, acacetin, epigenin, luteolin, epigenin, and acacetin, and different types of the molecules will be identified. That is the advantage of softwares presently used. That means artificial intelligence and learning system to discover the new molecules from the known molecules from the plant sources. Next slide. This is the statistical analysis studied by the scientists to what advantage of the databases to study the natural products towards the antioxidant capacity. Next slide, please. Then the last one is the feature of pharmaceutical pharmacognosy by integrating the rapid in vitro screening with high resolution chromatographic profiling, thereby unblock the true therapeutic potential of the natural matrices. This approach is unified approach is rapidly accelerating the development of functional foods, highly targeted nutraceuticals, and novel pharmaceuticals designed to restore cellular redox homeostasis. This is the today's brief discussion about on the antioxidant capacity and its present techniques used for studied the antioxidant capacity, that is the total antioxidant capacity by using HPLC, UPLC, by added advantage with the hyphenated technique like the mass spectroscopy. Thank you for your patience hearing my seminar. Thank you, sir.
Thank you, sir. Thank you very much. Audience, do you have any questions regarding the topic which has been discussed by Dr. A.S. Rao, sir? Sir, I have one question from YouTube, sir.
Tell me, sir.
Sir, the participant is asking for the estimation in pigmented fruits and seeds. You quoted HPLC, HRMS. Which is better, whether it is HPLC, HRMS or else HPLC, MSMS?
HPLC, HRMS is better, sir.
Why? Because in present days, there is the softwares are developed, which are HRMS attached. If you suppose, okay, so one extract given for the HPLC, it will be separated out on the basis of the method optimization. All the molecules are coded by the HRMS. Whereas MS by MS, resolution is the problem. It may be difficulty to identify the compounds because of lacking the good resolution. Whereas HRMS, because of high resolution, that means 20,000 units of resolution will be provided by the HRMS. Thereby, even the little bit cost is more, then HRMS is always better.
Sir.
Okay, sir?
Thank you, sir. Thank you for your valuable insights in antioxidant evaluation in herbal products. Thank you very much, sir. Now we have our guest speaker today. He's none other than Mr. Nagasuriya Prakash Padhala. He's working. He's now a director and research co-founder of BioLeaves. So just I welcome him for the speak. Good morning, sir.
Yeah, yeah, good morning. Am I audible? Am I audible to everyone?
Yes, sir, you are audible, sir. You are audible, sir.
How about the video? Am I visible to? Am I visible to?
Yes, sir. Yes, sir. You are visible, sir. You are visible, sir. You are audible also. Let me introduce to the gathering.
Okay.
Yeah, Sri Nagasuriya Prakash sir is now working with BioLeaves. He's a person of pharmaceutical analysis working in mass spectroscopy, and he's a scientist. Has a war and experience of 17 years in regulatory and non-regulatory bioanalysis. And he previously worked with Civil Life Sciences. As he started his career as a trainee, and now he became as a leading scientist in bioanalytical team of BioLeaves. He's not only a leading scientist, he also a co-founder and director of BioLeaves Research Center. So he has been through many discovery research portfolio, and he has a good team also. He worked with he maintained a good team. He provided with technical oversights in method development, method transfer, method validation, sample testing, troubleshooting of bioanalytical methods, whatnot. So he also had the bioanalytical exposure through metabolites, drug metabolites, biomarkers, and peptides in conventional complex biological matrices. So he has several research publications, and he is peer-reviewed and also in very peer-reviewed international journals. He has presented several international forums. Currently, he is leading bioanalytical research services at BioLeaves. Thank you, sir, for a great opportunity to have you in you as a guest speaker for today's webinar. Over to you, sir. The webinar is yours.
Yeah. Thank you, Mr. Kranti, for a quick introduction. So just before beginning this, it's the first of its kind for me to appear on such an conference or a virtual seminar, whatever it is. I just would like to understand what exactly it is and how frequently it is being conducted by you and your team, and how about the attendees who are all present. Can you just let me know on that?
Yes, sir. Actually, yearly once from the Department of Pharmaceutical Analysis, Institutional College of Pharmacy, we used to conduct this webinar, sir. As a part of this webinar, there are PG students who are going to attend it, UG students are going to attend it. And many people like some faculty, like professors, assistant professors, and associate professors will attend the webinar, and they will know the new things from the Department of Analysis, like today's talk.
Okay.
Yes.
Okay, good. Yeah. So I think I don't need any other introduction, to be given by myself. So, I'd like to jump quickly into the topic.
Yes, sir. They need to share the documents, sir.
Yeah, you try. You please share the the PPT or the PDF of the PPT that I have sent you.
It is on the screen, sir.
Yes, yes, yes. Yeah, it is visible. It is clear. So I thank once again Dr. Natraj, for inviting me uh, to take part on this, platform, trying to spread or the knowledge or the information what I have gained and gathered over the 2 decades of experience that I have uh, in the field of bioanalysis. So the topic that I was requested, to give a talk for about 30 minutes is, sample preparation or sample preparation methodologies or technologies that are used in um, quantitative bioanalysis. So I do not want to concentrate more on the sample preparation itself or the techniques itself because, you know, there are other variables or components which are equally important than the techniques or the methodology itself. And most of the graduates or the scientists, overlook, for such information and obviously fail at one point of time, ignoring some of the critical components and variables. So I have prepared a slide deck of about 5 slides, not just covering the sample preparation techniques, uh, not just the sample preparation techniques, and other important variables and components. So can we go to the next slide?
Thank you, Prakash, joining. This is Dr. Natraj. Okay? Thank you. Thank you.
Thanks a lot. Thanks a lot. Kranti, can we just go to the next slide? Yeah. So let us try to understand what basically bioanalysis is. So as everyone knows, it's nothing but the analysis or analyzing, something within a biological material. So it is, it can be said as a subdiscipline of analytical chemistry, which is used for quantitative measurement or determination of xenobiotics and biotics. As all of you know, that xenobiotics are nothing other than the chemically synthesized or naturally extracted drug candidates and genetically produced biological molecules and or their metabolites or post-translationing modified products. And biotics are something different. These are nothing but the macromolecules, which proteins, DNA, or the endogenous metabolites can be referred to as biotics. So, what is the role of bioanalysis in drug discovery and development? It's very critical because the focus of bioanalysis in the health industry is to provide uh, a quantitative measurement of active drug and its metabolites, or its biomarkers for the accurate measurement of pharmacokinetics, toxicokinetics, bioequivalence, bioavailability, and exposure response relationships. So, exposure response in the sense, pharmacokinetics talks about exposure, and pharmacodynamics speaks about response. And toxicokinetics, and similarly, toxicokinetics and toxicodynamics. So these are all required, to support the drug development and drug discovery and development, apart from post-approval TDM, which is nothing but therapeutic drug monitoring. So that is where the importance of bioanalysis lies with. And coming to the techniques, the quantitative techniques or the instruments that are available for this activity, or LC connected with UV or fluorescence detection, uh, GC gas chromatography connected with MS, and liquid chromatography connected with mass spectrometer. So and at this point, I assume some of you must be having a doubt that how GC-MS is used for quantitative analysis of therapeutic molecules or compounds in biological materials? Yes, it is very much important. And I can just refer a small example when it comes to GCMS or the application of GCMS. So there is a product which is called as iron sucrose. So it is available as an IV injection, I mean injection which can be administered through intravenous route. So uh, which is meant for iron deficiency anemia, and this is most prevalent in CKD, in patients suffering with CKD, chronic kidney disease, or in patients who doesn't have the ability to absorb iron, which is available in the bloodstream. So GCMS has, very strong role, particularly in measuring sucrose, because iron is available in two different forms, ferric and ferrous form, and sucrose is available as sucrose only. So the GCMS also has equally got its importance when it comes to bioanalysis. So among all the currently available techniques or the instruments, I would say LCMS MS is the most important thing and most widely used and the most reliable tool for measuring due to its high specificity and the sensitivity. So these are some of the advantages of using LCMS. And so, what are the common biological materials that are used to measure any therapeutic compound? So that can be broadly categorized into two: body fluids and organ tissues. So body fluids range all the way from whole blood, plasma, serum. It can be urine, so this tears, so which comes from your eyes. So it can be saliva, it can be the gingival crevicular fluid, which is taken from the gum, and it can also be synovial fluid, which is present in the joints. So the molecule itself, I mean the compounds itself are present in very low quantities or in very low volumes when it comes to certain areas which are a part of these biological fluids. I personally have the experience of working with all these matrices that I have referred. There are instances where only 2 microliter of a biological material would be available, for a bioanalytical scientist to measure. And one such example is the gingival crevicular fluid, which is available or collected at the gut. So the organ tissues, they also range from everything, skeletal muscles, your skin, so eyes, uh, so kidneys, lungs, and brain as such, or the different regions of a brain. For say, cerebellum, frontal cortex, thalamus, hypothalamus. So these are some of the most broadly used um, body fluids and the organic tissues. But the uses of these tissues, vary at different stages of the development of the molecule. So it is only at the preclinical stage or the basic research level where this entire broad array of body fluids and organ tissues are used. But when the molecule moves from preclinical to clinical, it is hardly 2 or 3 biological material which would be required to measure, the molecule of interest. So can we move to the next slide, please? So now, having known what bioanalysis is, then why can't we inject directly the compound into the instrument which is used to measure? So that is where the sample preparation exactly uh, steps in. So biological samples contain abundant, endogenous components such as salts, so sodium, magnesium, potassium, calcium, chloride, etc. Apart from small molecules are those which are endogenously present, and they which are less than 1,000 daltons, I would say. Some examples of that, small molecules are amino acids or carbohydrates. So these are And next, proteins and lipids. These are the certain examples for endogenous compounds. And apart from them, the exogenous components. So what are those exogenous components that are taken from outside? So API as such cannot be administered to a patient or to a laboratory animal which is meant for experimentation. So APA is given in a combination, which is prepared as a formulation which requires a lot of vehicles. So those vehicles can at times interfere with the analyte of interest or the molecule of interest. So that is where the biological sample preparation actually pitches in. So in simple, there are three parameters why a sample preparation is required. So it is for the isolation, purification, and concentration. So which again reinforces that sample preparation is nothing but a pre-analytical separation process that involves selective isolation of the molecule of interest from the biological material, because the biological material is found to be bound to proteins or several other endogenous compounds that I referred to you. So it has to be isolated from that biological material. And after that, followed by minimization or elimination of the biological material itself. Biological material we have to eliminate. So, which is nothing but purification. So, and ultimately, the last one is nothing but concentration. So, which can be referred as the enrichment of the compound. Because at times, certain compounds are present in very low concentration. So that depends on the potency of the molecule. So if the more the higher the potency of the molecule, the lower it is present in a biological material or a biological system. So, so this is the importance of having sample preparation. And an ideal sample preparation method must focus on two things apart from the isolation and all. So a sample preparation technique must be uh, capable enough to either minimize or reduce the effect of matrix, so which is contributed by the body fluids or the endogenous compounds or the exogenous compounds present in the body fluid, apart from maintaining a reasonable and consistent extraction recovery. These are the two critical parameters, the objectives of having a sample preparation procedure. And as most of you know, that the most commonly used techniques are, there are only three, and within the three there are again several sub procedures which can be referred to. So they are nothing but protein precipitation, liquid extraction, solid phase extraction, and I would brief you on these three probably in the last slide. Can we move to the next slide, please? So I would request everyone, all the viewers, to pay attention to this particular slide, which is nothing but the physiochemical properties. Because it is my personal experience that most often all the graduating students or the scientists during their earlier stages of the career would overlook and would always ignore the importance of these properties when it comes to developing a bioanalytical method. Because physiochemical properties are something, I would say it is the first and the foremost important aspect of sample preparation, LC-MS bioanalysis, because it helps us to understand, and utilize the relevant physiochemical properties of the molecule of interest. And these are included, but not just limited to hydrophilicity, lipophilicity, and proteolytic properties such as log P, log D. So understanding these parameters, is very important and help a bioanalytical scientist to choose the right and the most suitable preparation technique and the associated experimental conditions, for that particular molecule. So let us just discuss, for about a couple of minutes in brief about about these properties. So, as all you know, as all of you, most of you would be knowing the definitions of what is hydrophilicity and hydrophobicity. So, I would not get into the details of that. But what all I would say is, so there are two critical parameters which help us to understand the hydrophilicity and lipophilicity. Those two parameters are nothing but the partition coefficient, which is also called as log P, and the next one is log D. So these are the two important parameters that help us to understand what exactly are the hydrophilicity and lipophilicity and how they are in fact derived. So let us look into them actually. So partition coefficient, so which is nothing but the log P. So it is the concentration ratio of anionized form of the molecule which is present between two immiscible organic phases or two phases, two immiscible solvents. So one is the aqueous, the other is the organic, which are present at equilibrium. So this is considered, log P partition coefficient is considered as a measure of hydrophilicity or lipophilicity or hydrophobicity of a given molecule, for which the higher the value of log P, the more lipophilic or the more hydrophobic the molecule is. So there are certain workflows which actually, tells us, how these experiments, how the lipophilicity or hydrophobicity or log P has to be determined. So coming to distribution coefficient. So it is something similar to partition coefficient, whereas in partition coefficient, we discussed about just measuring the concentration of one particular form of a molecule, that is, which is present in anionized form. But whereas when it comes to distribution coefficient, so it is the ratio of sum of both forms of a molecule, both the ionized as well as the ionized form of the molecule between two immiscible solvents, which is nothing but aqueous and organic phases. So, these two are very, very much important. That's the reason why I've been explaining more in detail about this. And coming to the proteolytic properties, so this is something I don't say it is new, but it is a different term which is used very rarely. Proteolytic property, which nothing but refers to a process in solution where one proton is transferred, where a proton would be transferred from one molecule to another. So this directly refers to the intrinsic activity, or the ability of the molecule to either donate or accept a proton. So PK is nothing but I would say, a mathematical measure of the prolytic property. And here we have to understand the relationship between pH and pK. pH is equally important here because, as you all know, that in chemistry, pH is nothing but the negative logarithm of the concentration of hydrogen ion, and which is specifically used to define the acidity or basicity of an aqueous solution. So pK is nothing but, it is acid dissociation constant. pK can also be defined as the pH where a molecule exists in 50% as an ionized form and the remaining 50% as an unionized form. So, but many organic compounds or therapeutic compounds, they show these proteolytic properties and contain both acidic as well as basic properties. And pK of a compound can inform exactly the charge state of a compound at a given pH. It tells us how acidic or basic the compound is. When the pH is equal to pK, as I said, you 50-50. But when the pH is less than pKa, so which means the environment is more acidic than the molecule's pKa. So the molecule will be in protonated stage. It exists in the protonated form, indicating that it is ready to accept and hold the protons that it receives. So whereas when the pH is greater than pKa, the environment is more basic, indicating the compound or the molecule exists in deprotonated form, which is ready to donate a proton. So, so why all these properties are important? I mean, why the physical properties are important? Where they exactly play a role? They majorly play a role in defining the extraction efficiency. What we call is that the recovery of the molecule from the biological material. So, having a basic understanding on these properties would help a bioanalytical scientist to extract maximum amount of the compound or the molecule which is present in the biological material. So I would say these are some of the basic, fundamental principles that most of the graduates or the scientists would ignore. Probably that could be due to lack of knowledge, or that could be due to work on so the hello Kranti, can we move to the next slide?
Yes, sir. Your video just paused, sir. Now it is okay, sir.
Yeah, I happen to receive a call since I'm using my mobile, not to attend this.
Sure,
So
sir.
that's
No
fine.
problem. No problem.
Yeah. So these are few of the other variables which play a very critical role during sample preparation. They are nothing but the stability of the molecule and NSB, non-specific binding of the molecule, and protein binding and blood to plasma partition ratio, blood to plasma ratio or blood to plasma partition ratio. So stability of a compound in a given biological material is very, very important, because the more the molecule is unstable, the more the possibilities of that compound or molecule are to be either underestimated or overestimated. So certain preventive procedures or measures have to be in place before continuing with the sample preparation. So one should carefully examine. So in order to address this stability, one should carefully examine and understand the chemical and structural characteristics of the molecule, and the in vivo and in vitro transformation of that particular compound prior to developing the sample preparation. So how is that possible? Yes, that is obviously possible. So for NCs, it's altogether a different procedure where there are certain workflows of how that has to be executed. But when it comes to generic molecules, now there is sufficient evidence on the stability of all the available molecules in the market, of how the stability has to be addressed. So if at all there is not much information available on any particular compound or molecule of interest that you wanted to work with, so you can always try to gather information of structurally similar or molecules which are very much close to that. So this is the ideal practice that one follows. Generally, this would help bioanalytical scientists to understand some of the potential stability alerts or the instability alerts. So because the for a bioanalytical scientist, each molecule is unique. It is different. So one has to understand this. There are several molecules, there are several molecules within, though they fall within the same pharmacological class, but they differ, because of the presence of various functional groups or the chemical groups. And some of the groups that pose a great challenge to bioanalytical scientists during sample extraction or preparation procedure are, lactones, amides, esters, thiols, and catechols, and several other. So these are some of the functional groups or the chemical groups which are prone to degradation So, and there a lot of importance have to be given while handling or working with these molecules. So, next coming to the NSB. So, what is this NSB? Non-specific binding. So, this is, I would say, it's inherent uh, ability, unintended. I would say it is unintended intrinsic property of the molecule to adsorb or bind to the surface of the container. That could be either glass or that could be either a polypropylene plastic. Uh, that can be as simple as a pipetting tip, which normally the bioanalytical scientists would use to dispense the biological material. So this NSB, this phenomena is majorly driven by electrostatic, the charges and the lipophilicity or the hydrophobic uh, properties or the behavior that exists between the sample and the container. So this is very much important. So if we consider where in which particular biological material this matrix or this NSB would be observed, I would say it is urine and CSF, because if you look closely and try to understand, so majority of molecules or compounds, they have high affinity to bind to proteins. So when such compounds have to be measured in biological, material that are devoid of proteins. So materials which does not contain proteins are, urine and CSF, cerebrospinal fluid. So so such matrices mostly poses a challenge, and that can lead to non-specific binding, which causes invincible loss of the uh, drug, leading to poor recovery, which can lead to inconsistent assay results and inaccurate measurements. This ultimately results in negative results, false negative results. And these are also concentration dependent because NSB is not consistent. So NSB is more can be observed at low concentrations of a sample when compared to the high concentrations. So which is something called concentration dependent skewing. And it also leads to carryover because when a compound has some exhibit NSB at a higher concentration, so there always would be some concentration left from higher concentration, which keeps on carrying to the. So it also has to be addressed. So there are certain procedures which can, which can mitigate this NSB. So some of the measures that have to be taken while addressing this NSB uh, is the coating the material. There are certain detergents which are used to coat the glassware or the pipetting devices or the polypropylene material. Or I know there is something called BSA, bovine serum albumin. So this is a type of blocking agent which can bind to all the sites that are active and open, within the container system. So these are some of the strategies which can be used to mitigate NSB. And the most class of compounds which are prone to this is the large molecule. It's not the small molecules which are prone to NSB. It is the large molecules such as proteins, peptides, oligonucleotides. So this is one area bioanalytical scientists have to pay attention to. So next, coming to the protein binding. By the way, do we have time? I think it's already 12 past. Is it okay? Uh,
Yes, sir. We have, sir. We have.
yeah, I would just need only 5 to 10 minutes of the
Sure,
additional time.
sure, sir. Sure, sir. Sure.
Yeah, protein binding. So as I said you earlier, any given drug or any given drug molecule generally exists in two forms. One is the bound, the other is the unbound. and that could be either in blood, plasma, or serum. So if the protein binding is reversible, then a chemical equilibrium will exist between the bound and the unbound states of the drug molecule. So if you take albumin, so albumin is it's a type of plasma protein and it is basic in nature. So since it is basic in nature, the more active and neutral drug molecules will primarily bind to albumin. So even every protein has certain sites, limited sites, I would say. So when those sites are saturated, which means the available sites are being bound, by the acidic or neutral molecules, the available acidic and neutral molecules would try to find, another albumin, or I would say, see the other area, such as lipoprotein. It's also a kind of protein which is basic in nature. When albumin sites are completely saturated, the remaining acidic and neutral molecules would try to bind to lipoprotein. So it's the same case with basic drug molecules also. Basic drug molecules, they show the affinity to bind to one particular glycoprotein which is called acidic 1 acid glycoprotein, which is acidic in nature. So these proteins are, they are present in, they are part of, whole blood, plasma, or serum. So therefore, the release of the analytes or the molecules from these plasma proteins is very much important. How that has to be done? We have to interrupt that binding, and it's a key step for good and consistent, recovery. I would say, regardless of the choice of sample preparation method or the protein binding rate of the analyte. So in generally, so what one would do to address this protein binding is uh, the biological materials are generally treated with either acids or bases or with a buffer solution of certain pH or with an organic solvent to disrupt this binding so that the analyte would be released from the site, from the plasma proteins prior to further processing. So this is also one other component or variable which is equally important. So the next one is blood to plasma ratio. So as you all know that it's nothing but the ratio of concentration in whole blood containing both RBCs plasma to the corresponding value in plasma. So it's equally important for molecules that are intended to be measured in whole blood as well as plasma. Because there are certain
we have a problem with your audio, sir.
Who are based damning plasma. Everolimus. So everolimus has more than 65% affinity to bind to RBC rather than to plasma.
Sir, excuse me, sir.
So it is very important when someone with such compounds have to take such precautions and have to aware of a phenomenon which is called temperature equilibration. So if a drug molecule between BC plasma, this can impact the measurement of that molecule with the intended. Suppose when someone is working with the blood, the ideal temperature that sample has to be stored is 2 to 8. It cannot either be stored at freezer temperature, which is -80, and it cannot be. It can neither be stored at. The more we try to store whole blood sample, which consists of a molecule, which exhibits this phenomenon, this temperature dependent equilibration. So it leads to false negatives because molecule will be gradually moving RBC to the plasma. So this is one other important aspect.
Sir, your audio is a bit low, sir.
So can we go to the last slide, which
Sir, your audio is bit low, sir. Audio is low, sir.
The most commonly used sample preparation techniques.
Sir, your audio is low,
So I
sir.
would say this is the last one because before coming to this stage, one has to focus majorly on, the earlier components or variables that I have taken involved. So as everyone would be knowing, there are widely three techniques that are used. So protein precipitation, liquid extraction, and solid phase extraction. There are, equal advantages when it comes to talk about protein precipitation. Protein precipitation is one of the most simple, quick, uh, uh, which has high throughput and high recovery. Because it doesn't require any other solvents or any other complex. One would not be requiring the pKa or log P or log D, as I mentioned earlier. So it can also be referred to as simple dilution technique, where the plasma would be trapped, would be diluted with 3 times or 5 times of its volume with an organic solvent, which can be either acetonitrile or methanol. But this is quick, as I said, this quick, simple. This is mostly used in, by the biological scientists during the discovery and development phase. But when it goes to the human clinical phases, this is not the ideal technique that is mostly used because it has its own limitations or drawbacks, I would say. Because this technique doesn't remove the exogenous or endogenous components, matrix components particularly. So obviously they can interfere during analysis, particularly during ionization process of the mass spectrometer. It can interfere with the molecule of interest. But again, I would say it is absolutely possible to overcome that as well by adapting to the elution technology. So as you all know that there are two types of technologies, isocratic and gradient. So gradient is one technique which can overcome the challenges, that when a protein precipitated sample analyzed by isocratic method uh, can overcome the challenge. Isocratic method, gradient program can overcome all the challenges because the gradient program has to be developed in such a way where all the phospholipids or proteins which are present in the solvent or the final extract are eluted at a different RT retention. So coming to the LLE. So the principle involved in LLE is partition coefficient. Is again, as I said, you for log P and log P, the same principle log P, which is the same principle which is involved here as well. So the distribution of the analyte between two phases is related to its either log P or log P values. So by changing the organic solvent or by adjusting the pH and buffer ionic strength, the target analyte can be favorably extracted from the aqueous phase into the organic phase, leave most of the matrix components such as phospholipids or proteins or organic or salts in the aqueous phase, inorganic salts in the aqueous phase. But there is one major limitation for this technique. So that is nothing but it cannot be used for compounds which are more hydrophilic in nature, which are more polar in nature. It is possible, with the selection of certain chromatographic columns, but I would say it is definitely very resource intensive to develop a method, particularly using LLE for hydrophilic or polar molecule. So the final one is the solid phase extraction. So this is one of the best sample preparation technique or methodology when we compare to the rest of the two techniques, because it provides a very clean sample. So which can provide a very clean sample. and large volumes of samples can also be used. And the principle in this particular technique involved is nothing but the absorption. So absorption. So this is based on the differential affinity. So sunlights between a liquid phase, which is nothing but a sample, and a solid phase, which is nothing but a sorbent. So the sorbent is placed uh, inside something called a cartridge. It consists of a sorbent, which utilizes selective absorption and elution to isolate or concentrate the target components. This is, I would say, this is technically a low pressure chromatography. This is one form of low pressure chromatography. So, like for LC columns, there are the different columns which are available with different chemistries. So, similar to that only, the solvents which are used or the cartridges which are used for solid phase extraction technique also consists of several, chemistries. So, if you are, I just would like to cite few examples for that. One is called HLB, hydrophilic lipophilic. So the other is weak cation exchange, strong cation exchange, weak anion exchange, weak cation exchange. But how you choose these categories all depends on the uh, chemical characteristics, log P and log D values of the molecules. I think, and the limitation that is this particular technique is very expensive because the cartridges are highly expensive, and the sourcing of cartridges is also time taking, time consuming, and it also requires certain level of understanding of the science by the scientist or the technician who actually performs that, and it certainly requires some skill when compared to the two. So these are simple workflows where a defined protocol would be there for the scientists to execute. And these can be learned, once someone try to enters into the industry. But it doesn't help you unless help you in gaining the confidence unless you have some understanding of the physiochemical properties and the other variables such as stability, NSD, so on. So I think I'm done with this, and thank you so much.
Yes,
Thanks
sir.
everyone for your attention.
Thank you, sir. Thank you. We'll ask audience for the doubts, sir. Audience, do you have any doubts? Please post in the YouTube group or else post in the this
Hello,
one. Sir, am I
hello.
audible? Sir, am I audible, sir?
No, Kranti. You're not audible.
So my audio is on. Can you please see? I think
Hello.
you have
I
muted
think
me.
I lost something
Yes. You have muted me, sir.
to me.
Maybe you have muted, sir.
Yes. And now I think I can hear you.
Yes, sir. Now it is good. Sir, we will ask audience about any doubts for just 1 minute session. Yeah.
Yeah.
Yes. Audience, do you have any doubts? Please post in the text, either in the YouTube or else in the Zoom. Sir, already we have one text, sir, one doubt. So they texted that.
Yeah, your voice is very low. Your voice is very low.
Sir, we have one doubt from the YouTube, sir.
Yes.
Yes. They are asking, do we have any advanced green extraction techniques? They are asking, sir. Which is
Green extraction techniques.
suitable to practice in academics.
Green extraction techniques.
Yes, sir.
No, green extraction techniques are something which doesn't involve the uses of any sort of organic solvent. I don't think so. There is any sort of technique which is currently available.
Sir. Sir, another question, sir. Do we have any nanomaterial based sorbents for extraction? Can you name them if it is marketed?
Nanomaterial.
Yes, sir. Nanomaterial based sorbents, sir.
I happen to work mostly with the sorbents that are designed and developed by Waters, which is a very well-known US-based company. But I have never come across. They must be available. I'm not that much familiar with that thing.
Yes, sir. Yes, sir. Sir, there is another question, sir.
Mhm.
Can you explain about micro extraction by packed sorbents, sir? Packed sorbents.
Micro extraction by?
Packed sorbents. Packed sorbents.
No, can you spell it? I couldn't get it. Micro extraction can be anything, where where only very small volumes. As I referred earlier during my presentation, I said something about the body fluid, which is nothing but the gingival crevicular fluid.
Yes, sir. Yes.
Or there is one more technique which is called as dried DBS, dried blood spot.
Yes, sir.
No, it is a card, dried blood spot.
Yes, sir.
The students or the graduates can refer to this dried blood spot, where instead of using this particular the significance of this dried blood spot technique is, you know, certain trials involves the use of adolescents, infants. Okay. So in such a cases, it's not ethically permitted to draw large volumes of blood samples from the infants or the adolescents because there are certain medicines which are available for infants and adolescents. So in such cases, very small the volume of blood would be collected just by pricking the finger, and they would hardly collect 2 microliter of the blood sample, and they just drop it on the paper which is present, which is part of the dried blood spot. So they are also available in different chemistries, and it obviously falls under micro extraction technique because you don't have much larger volume of the sample to process and to measure the analyte.
Yes, sir. That's all. Thank you, sir. Thank you very much for your valuable time. So, on behalf
Yeah.
of the organizing committee, I would like to propose a vote of thanks for the today's insights on the webinar, Analytical Insights to Antioxidant Evaluation and Bioanalytical Sample Preparation. First of all, I want to thank our guest speaker today, in spite of his busy schedule. I came across him because he has no time even to lift the phone, but still, he has given nearly about 1 hour of his time to give or else to deliver the very important, vital data about the bioanalytical samples and sample preparation. Sir, we are very much enlightened, sir. Our analysis students, PG students are very happy. And also the inputs you have given in the industry, that was very worth, sir. So thank you for your time, sir. Thank you very much for your time. And I also thank our speaker, HOD, Dr. A.S. Rao Garu, sir, for giving the antioxidant evaluation speak. And also I thank management, principal sir, and all the supported staff for organizing this webinar. And this was my honor to introduce Mr. Surya Prakash, Naga Surya Prakash sir, who is a co-founder of Biolist. Sir, we are very much warm for your speak, sir. Thank you very much, sir.
Yeah, I welcome all of you, and I appreciate all of you for inviting me to share some of the knowledge on this platform. Thank you very much.
Thanks to all participants who participated. Please fill the feedback link to get the certificate. Thank you very much. Thank you very much, sir.
Thank you. Bye bye.
Bye, sir. Bye.